Welcome back. We are going to take a look at the major accessory organs for the digestive system here. So we're going to look at the liver, the gallbladder, and the pancreas. Start with the liver. It's huge, largest organ in the body. It's got 4 lobes, and remember that it's anchored to the stomach via that lesser omentum. We've got a porta hepatis on the posterior side that allows the hepatic artery, hepatic portal vein, and the hepatic duct to come in and out of the liver. It's this duct that's going to fuse with the cystic duct and then become the bile duct. So the major digestive function of the liver is to make bile, and so that's how it's going to move out of the liver there. The liver is known for kind of having this high capability of regenerating. It's not a given. The liver cells, hepatocytes themselves, have the ability to regenerate if they are properly stimulated. But if the hepatocytes are damaged enough by cirrhosis or, basically what is a scarring of the liver, then they lose that capability. So, it's kind of a myth that you can just grow a new liver. It's not quite that simple. And so, you kind of want to take care of yours. One of the major things that, we're looking at in terms of damaging the liver is, drugs and alcohol. So it's not necessarily a digestive capacity of the liver, but one of the things that it does is it helps break down toxins from drugs and alcohol into something that can be secreted. And so that is one of the major contributing factors to liver disease and the need for liver transplants and things like that. Okay, so here's our right and our left lobe. We have a ligament, the falciform ligament, that then turns into the round ligament that runs through. And then on the posterior side, this is our porta hepatis right here. The infravena cava is running. There's a sulcus right here. This is the caudate, and this is the quadrate lobe. The gallbladder sits on the posterior side here, and there is that bile duct I was talking about. That's the cystic, and then that's the bile duct. Okay, if we look at the liver in a microscopic view histologically, we're going to see that it's divided into these kind of somewhat regular shapes called lobules. We're going to have the liver cells, or hepatocytes, all sort of radiating out from what's called the central vein of each lobule. And then at the corners of these sort of hexagons, we're going to have what are called portal triads. We're going to have a branch of the hepatic artery, a branch of the hepatic portal vein network, and then a bile duct that is going to exist there. Between the hepatocytes are the liver sinusoids. So remember that sinusoids are a type of capillary where we have a very irregular structure of the endothelial cells and incomplete basement membrane, and it's where we're going to allow large things to move in and out of the blood. So we've got sinusoids running all through the lobules that then lead into the central vein. And because we've got sinusoids, we're going to need a fairly significant immune presence, so we've got our hepatic macrophages there to help us out. The hepatocytes are going to do all this stuff. They're going to synthesize bile. They're going to secrete that bile into the little bile canaliculi that are eventually going to end up in the bile ductules, and then in a bile duct, and then out the liver and into the small intestine. They're also going to remove glycogen and amino acids and fat-soluble vitamins from the blood that's coming out of the stomach and the small intestine. And then we've got that detox that I was talking about. So the bile is the major digestive function. Bile is it's kind of a yellowish greenish color, and it's going to contain bile salts that's the component that's going to help us with our lipid digestion. But there's a bunch of other stuff in there too. A lot of cholesterol and triglycerides, phospholipids, electrolytes, pigments. Those pigments exist from the breakdown of red blood cells. And the lipids, that's one of the only ways to remove lipids from your body is to secrete bile into the digestive system. And then they're going to be removed with the fecal matter. All right, so let's take a look at this concept here. So this is a lobule that we would be looking at here. So here's kind of our hexagon, if you will. This is the central vein, okay, and these are the hepatocytes. These are the sinusoids you can see here. So here's our endothelial cells. There's a macrophage and red blood cells. And then at the triads, we've got a branch coming off the hepatic portal vein, a branch coming from the hepatic artery, and then a bile duct. So let's take a second right here and talk about blood flow. Blood coming out of the stomach and the small intestine is going to have things that we've absorbed in it. So things like those alcohol, amino acids, vitamins, nutrients, glucose, all of those different things. That blood does not immediately move into the venous supply and head back into the inferior vena cava and then to the heart. Instead, the blood coming from the stomach and the small intestine is going to move into what's called the hepatic portal vein. It's going to take this very nutrient-rich blood into the liver. So if you remember back to studying your arteries and veins, your APR images, that was the vein that was coming into kind of almost like the inferior, it's what it looks like, but the inferior portion of the liver. It's bringing blood in from the digestive system. So that portal vein is going to branch into this network that's going to end up with these guys right here. Now this is all venous blood. So oxygen's been dropped off, but it's got all these nutrients in it. So that venous blood is going to end up in these ductules here, or these venules here. And then you've got oxygenated blood coming in from the hepatic artery. And those two bloods, so this deoxygenated nutrient-laden blood and the oxygenated arterial blood actually mix and then flow through the sinusoids to the central vein. While you flow through here, we're going to remove the oxygen from the arterial blood, and we're going to pull out things like glucose and amino acids and vitamins that are stored in the liver from that nutrient-laden blood coming out of the stomach and the small intestine, so that by the time the blood feeds into the central vein, it's going to be deoxygenated and the nutrients have essentially been removed. The central veins then all connect together in a network that leads this venous blood out of the liver into the hepatic vein, which feeds into the inferior vena cava. So we have this network where we're going to take the venous blood coming out of the stomach and small intestine, shunt it over to the liver, remove all the stuff from it, and then back into the venous circulation. So you've got blood flowing in this direction in your lobules. Now the hepatocytes, you can see all these green things here. The hepatocytes are busy also making bile. And so all running through here are these bile canaliculi. Blood is flowing this way. The bile is flowing out towards these bile ductules, which are then going to eventually fuse into what are called the right and the left hepatic ducts, and then into the common hepatic duct, and then eventually through this ductwork that we'll talk about in a second. So anyway, blood is flowing this way, bile is flowing that way. So let's talk about these ducts. So here's my, my right and my left hepatic ducts that fuse to form the common hepatic duct. And then you've got the cystic duct that leads into the gallbladder. And then below the level of the cystic duct is what's called the bile duct. And that comes all the way down, runs through the pancreas, and opens up into the duodenum. So what's been removed here is the liver, because that would be sitting here, and the stomach, which would be sitting here. Okay. So the gallbladder's job is going to be to store the bile that was made by the liver, but isn't immediately needed to help with lipid digestion. So what happens is you secrete bile and you fill up this whole duct. And then the liver is consistently and constantly secreting more and more and more and more and more. And so it begins to back up into the cystic duct, into the gallbladder. The gallbladder's job is to store it and concentrate it. And it's going to concentrate it by actively transporting sodium out, which is going to allow us to reabsorb water, concentrating the bile. Now, sometimes when this happens, the concentration, the gallbladder makes stones, okay? And that happens when we reabsorb too much water, and usually in conjunction with having too much cholesterol and fats in the bile. So remember that the bile is how we get rid of these things. So if you have too many in your body to start with, you're going to start moving more and more and more cholesterol and triglycerides and things like that into the bile. And then when you begin to remove water, those are hydrophobic, and so they glom up basically, and they form gallstones. And so gallstones in and of themselves are painful but not a problem. The problem with gallstones is way down here. The bile duct shares an opening into the duodenum with the pancreatic duct. That is where we end up taking gallbladder out and how he ends up at art school and all of that other fun stuff. So we're going to talk about the pancreas here, and we'll look at how these gallstones are going to affect this. So pancreas kind of has its head sitting in that curve of the duodenum, and the major job of the pancreas is to produce all of the enzymes we need to break down all of your major macromolecules. Basically, we can't properly digest any of our nutrients without pancreatic juice. I mean, we get a little bit going on with the stomach, but without pancreatic juice, we can't break our food down into small enough pieces for the intestinal enzymes to work. So we have a significant issue if we don't release pancreatic juice. So we secrete somewhere in the neighborhood of maybe about 1.5 liters a day. It's basically water, those enzymes, and electrolytes. And the electrolytes are primarily bicarbonate. Pancreatic enzymes need a pH of about 8 to properly function. So let's picture, let's kind of rewind. We've got chyme coming out of the stomach. That's a pH of 2. Coming into the duodenum, and that's the primary thing that's coming into the duodenum, aside from some intestinal juice that'll have a little bit of bicarbonate, that's not going to be enough to get the pH from 2 to 8. And so we secrete a huge amount of bicarb from the pancreas to bring the pH up high enough that these pancreatic enzymes can work. Now, back to the relationship with the gallbladder here. The issue with gallstones is that they can block the release not only of bile, but of the pancreatic juice because they share that opening. That can lead to a condition called pancreatitis, where the pancreas actually goes through a process of autodigestion. If your body is calling for the release of these chemicals, we've got the bile and the pancreatic juice, and we're going to talk about the release here in a second, but if the signals exist and we're saying do this, do this, do this, but there's a physical block in releasing them, what happens is that the pancreatic enzymes become activated in the pancreas themselves, and you begin to auto-digest the pancreas, and it basically shuts down digestion. altogether. So here's a couple little comics that kind of show that. These are funny little comics for something that is actually a pretty significant and serious issue. So that's really the problem with gallstones. In and of themselves, they're painful, but they're not life-threatening. It's their effect on the release of pancreatic juice that causes us to have to remove the gallbladder if we can't get them under control. Okay, so let's talk about this regulation, the secretion, basically. We have two hormones that are going to be released in response to chyme hitting the duodenum that are going to stimulate the release of bile and pancreatic juice. So when you get the movement of chyme into the duodenum, you're going to have pH receptors that are going to be activated, and they are going to cause the release of a hormone called secretin, which is in yellow here. Secretin is going to travel, and it's going to cause the release of bicarbonate and pancreatic juice from the pancreas. The lipid content of the chyme is going to cause the release of a different hormone called cholecystokinin, or CCK, that causes the gallbladder to contract and release that bile that's going to help us emulsify lipids. Now, both of these hormones can affect the other thing. So secretin has an effect on the liver and the gallbladder. Cholecystokinin has an effect on the pancreas. But what I've given you here is their primary effector. So the easiest way to kind of separate them out is to think about their major effect. Secretin targets the pancreas more than the liver and gallbladder. Cholecystokinin targets the liver and gallbladder more than the pancreas.